Okay, we're going to get started. My name is Simon Vukelj. I am the Chief Marketing Officer of the Children's Tumor Foundation. Welcome to this May is NF Awareness Month webinar on gene therapy in neurofibromatosis. Before we get started, just a couple housekeeping rules. First, this webinar is being recorded and we will post it online probably early next week. It'll just give us the chance to caption it properly and it'll be available to everyone for reviewing and sharing. Everyone is already muted if you're not a panelist, but if you are unmuted at some point for the Q&A, please make sure to mute yourself again afterwards. After this webinar is over, if you're ever looking for more information about the Children's Tumor Foundation and the work we do, that's available at ctf.org. You'll see that address on the screen. Very importantly, for the hearing impaired community, closed captioning is available. There is a CC button at the bottom of the screen. Please click that button and you will be able to adjust the size of the captioning. If you have any questions or challenges with that, please share that through the chat function and we'll certainly try to address that. And similarly, if you have questions through the Zoom chat, please submit that in the chat function or raise your hand. We'll probably take questions more towards the end. We're going to let our speakers get through presentations, and then but feel free throughout to submit your questions and we'll start getting ready for the Q&A section. With that, I'd like to turn it over to Annette Bakker, the president of the Children's Tumor Foundation, who will tell us what we're here for today. Annette, you're on mute. We're not hearing you.
Thank you, Simon. Hello, everybody. Over the past few years, we have received a lot of questions around the value and the opportunity of gene therapy for neurofibromatosis and for the NF patient community. As we all know, there have been a lot of treatments that have been evaluated and quite a few even made it to approval. These gene therapy approvals have obviously led to an increased appetite for gene therapy for many rare diseases. And gene therapy to all of us really feels like that holy grail. And that is why today it is my absolute pleasure to introduce a very distinguished panel of experts. Dr. Peter Marks, who is an MD, PhD, and is the director for the Center of Biologics Evaluation and Research at the FDA. Dr. Peggy Wallace, who is PhD professor molecular genetics and microbiology at the University of Florida College of Medicine. And she's also one of the key players in the discovery of the NF1 gene. And Dr. Scott Plotkin, MD, PhD, Armenise Family Harvard Professor of Neurology, also the chairman of the Clinical Care Advisory Board of the Children's Tumor Foundation, and a big believer as well in NF gene therapy, as he is also the co-founder of NF2 Therapeutics. So we felt that there would be no better panel today than these three experts to give you a better understanding of the NF genes and the opportunities and also the limitations of gene therapy. So with this, I would like to turn it over to Professor Peggy Wallace, who, by the way, is also the Young Investigator Award Chair of the Children's Tumor Foundation. Peggy, take it away.
All right. I share screen here. Okay, hello everybody. I'm Peggy Wallace. Again, I'm a professor at the University of Florida, and I've been doing NF1 research for over 30 years now. It was part of my original training at Michigan, and have continued to do that work here at the University of Florida as a faculty member. So I've been focusing on neurofibromatosis type 1 or NF1, and I'm going to consult with this Children's Tumor Foundation on some gene therapy pilot projects as we start to look into the possibility of gene therapy in neurofibromatosis type 1. So I'm here to describe to you what this would involve for NF1, and again, as Annette said, the promises and the pitfalls. So everybody has 2 copies of the NF1 gene in their DNA, as you can see over here on the left. You have 1 copy from your dad and 1 copy from your mom. This is a large gene on chromosome 17. And this gene, represented by the blue box here, is the blueprint to make a protein called neurofibromin, represented by the red box, and it has to go through an intermediary called the RNA. And so here we have the DNA, goes to RNA, goes to protein. Now, this protein called neurofibromin is very large, and we don't completely understand all of its functions, but we know it's important because all tissues express neurofibromin throughout life and throughout development. We do know that one very key role is to act like the brakes on cell division. So our body strictly controls when our cells divide all throughout our life. So this little car drawing down here, a rendering of a Lamborghini my son drew a number of years ago, represents a cell, and when it divides, it's like the gas pedal being pushed on a car. It's moving forward and it's dividing. The neurofibromin is like the brakes on the car. And there's front brakes and rear brakes. And you can consider it as if the front brakes came from the mom and the rear brakes came from the dad, for example. It's because you have two neurofibromin genes, and so therefore you've got two opportunities to make neurofibromin. So there's a very, very precise balance between cell division, gas pedal go, and neurofibromin brakes. Neurofibromin isn't the only brake in cells, but it's an important one in many cell types. So in NF1, one of these copies has a mutation in every cell, and that's represented by the X here. And whether it's on the mom's copy or dad's copy doesn't matter. However, what it represents is that one of the brakes is out, right? So for example, here the front brakes are out. Now, that's okay, like with a real car, because you still have your rear brakes. They're functioning, you can still stop the car. So cells in a patient with NF1, which have one mutated copy, which hurts neurofibromin function or eliminates it, is not going to hurt the cell in general. It's going to behave fairly normally. But you can have a random DNA mutation in that cell that then ruins the remaining copy with this green X. And that individual cell now is not making any neurofibromin. And since both sets of brakes aren't working, there's nothing to slow down the cell, and it is going to go and divide more than it should. So we call these cells two-hit cells because both NF1 genes are mutated. And these are the culprits. So for example, if both NF1 gene copies are mutated in a Schwann cell, it can lead to accumulation of too many of these Schwann cells, which are involved in nerves. So these Schwann cells accumulate in or at the ends of peripheral nerves. That means on nerves anywhere from where they start branching off the spinal cord to the ends in the skin. And there's a picture here of some neurofibromas that have formed off of the nerve ends on somebody's skin. And so these, if you cut them open, you'll see that there's simply too many Schwann cells there. The Schwann cells aren't all that abnormal, but there's too many of them there. That's why these are benign tumors. But they can cause a lot of trouble, especially with the ones that are in deeper nerves. If the cell that has the two hits is a pigment-producing cell in the skin, you'll get a cafe au lait spot. This generally happens during pregnancy and in the womb, as well as early childhood, because usually people have most of their cafe au lait spots by the time they're 3 years old. But certainly one cell had the somatic mutation, and then as it divided, it's making too much pigment as a result of not having neurofibromin working right. And then you get this spot. It's simply a birthmark. Fortunately, these are have no physical problems associated with them, but it is one of the signs that doctors look for to make a diagnosis. So the bottom line is that a mutated NF1 gene is the original source of the problem in neurofibromatosis type 1. Now neurofibromin, again, here's the gene to the protein, is again like a gas pedal, but what that does is it controls a lot of downstream pathways that control the biochemistry of how the cell grows and works and behaves. And so it's an important control protein. And this is important for normal cell and tissue function. when there isn't enough neurofibromin or it's not working right, these pathways become imbalanced. And that can cause problems like we talked about with accumulation of some cells or other problems also. And so until recently, the treatment approach has been to try to find drugs that can target these downstream pathway imbalances. And one of the big successes that just got it, the first drug that got FDA approved is the MEK inhibitor, Koselugo, and the MEK is, for example, one of these arrows. So MEK protein is involved in cell growth and division, and by being able to find an inhibitor that would work in the clinic, we've been able to come up with a drug that is ready for FDA approval for children for who have larger tumors. So this is a very exciting development. However, it doesn't seem to work quite for everybody. And there's some concern that if people stop taking this drug, that the tumors can grow back. And so we still need to develop more approaches to try to fight NF1, in part because it's so complicated. And we know it's not too likely that any one single approach is going to work perfectly for everybody. So because of recent advances in genetics, it's become possible to try to target the actual problem, the gene itself, instead of going to downstream pathways. So this is being pursued now, as Annette said, for a number of genetic conditions, since we do have new and better tools in recent years. And actually there's one FDA approved gene therapy already for a type of blindness that seems to work quite well. So I wanted to talk about the types of gene therapy that would be amenable to using in NF1 so that you can understand a little bit more about what the promises and what the limitations are. So one approach might be to actually add a new NF1 gene. So you have in the gene where both copies are mutated, they don't have any normal functioning gene. So what if you put a good one back in place? So that would be great. You could put a good gene in, it could restore normal neurofibromin function, the cell should go back to being normal. But there's a number of pitfalls with the technology we have at the moment. One is that the NF1 gene is just simply too big. It's way too big to be able to package and get into cells. But one option is to potentially try a smaller piece of the gene and see if that will work well enough. Another pitfall is if you put in this gene, it's not going to be at the same place as the original genes, and it might not produce the right amount of neurofibromin, and that might be very important to the cell. So that's another consideration that researchers have to take in terms of understanding whether that approach is going to work. And so those are some of the pitfalls that are being encountered when people are starting to consider research in gene addition, putting an NF1 gene or a mini NF1 gene in as an approach. Another gene therapy, gene-based type of therapy is gene repair. You might also hear it called gene editing. This is very new in genetics. It's only been around for about 5 years, and there's a lot of advances that are being made very, very quickly in this particular area. It has the promise to actually fix the original gene in its place. And that is, of course, exciting because then you don't have to worry about producing the right amount of neurofibromin. The gene will function the way it was always supposed to. But the pitfalls are that it's not a perfect system yet. It doesn't always make the repairs, and there is some chance of making errors instead. And so that has to be worked out. And also it may become necessary to do multiple versions of this type of therapy because these are actually based on either specific mutations or mutations in general that have to do with a particular region of the gene. And so these are more specific types of tools that we would be developing. The other approach that has to do with targeting the front end of the problem, the gene, is what I call evade the mutation, which is where you have some sort of drug, which can be an actual drug like aspirin, or it could be a DNA type of drug. And what you do is these are designed to trick the cell into ignoring the mutation that's on one of the gene copies. And so The premise of that is that you wouldn't have to add a gene back, you wouldn't have to edit the one that's in place. The pitfall is that you have to be able to get it to work well enough to stop the symptoms. It also is going to be a multiple therapy type of situation based on the mutations, and it's not permanent. This will be a therapy that you would take regularly because it's not actually fixing the gene. Usually these have more to do with how the gene functions or the RNA. So there's some additional considerations to be considered in research for gene addition and the gene repair editing. So these are the considerations that apply not to just NF1, but all of gene therapy approaches and all of the different kinds of conditions. First, you have to show that you can develop a therapy and get it to work well enough. You have to show that you can get the therapy into enough cells and the right cells in order to be able to create the effect that you want. You have to consider, are the effects going to be different on normal versus tumor cells? So cells that only have one mutation versus cells that have two mutations. Is the therapy going to have to be repeated if needed? And can you design it in a way that it could be repeated? So how permanent could this therapy be? And could you repeat it if you needed to? People are also considering gene therapies that are going to be specific to certain tumors. Maybe you don't need to treat the whole body, but there's one tumor that's causing a lot of trouble, and you can design a therapy specifically for that tumor, or use one of the tools being developed now and actually target that specifically and eliminate that problem. You might also want to think about, we've talked a lot about tumors, but targeting other features besides tumors. So gene therapy can be designed to go into different cells and try to correct different things. But above all of these approaches have to consider safety, as with all research aiming at developing new therapies. So not only do the scientists have to come up with the ideas and implement those into actual experiments and show that they can develop the tool and that it's going to work well enough, but it's got to be safe. And that's where it goes over into the clinical direction. So in the long run, to treat NF1, we may need multiple therapies, both drug and gene-based, because there's so many different mutations and so many different clinical problems in every individual who has NF1. And so it's a complicated condition, and it won't surprise me if that's what we need to have, is a toolbox full of multiple tools that we can pull out the right one for NF1 for that particular individual or family. I want to mention that the Children's Tumor Foundation is funding 2 pilot grants for initial experiments in gene editing in NF1. It's never been done before, and so these are kind of groundbreaking experiments. We selected 2 two-year grants to fund out of 6 applications that came in, 1 in the US and 1 in Australia. And they're testing approaches to do gene editing and to some extent gene replacement in Schwann cells. So we're starting to get in that direction through this Children's Tumor Foundation, and there are other foundations as well, such as the Gilbert Family Foundation, which are also starting to fund some new directions in gene-based therapies. So I'm going to hand it over to Dr. Scott Plotkin now, who can tell you about these approaches in NF2. Let me stop sharing. All right.
Thank you,
Scott, you're on.
Dr. Wallace. Can you hear me?
Yes.
You can. Okay. Thank you very much. I'm going to try to bring up my slides real quickly. I see them here. Okay. If someone could confirm when they see something, that would be helpful for me.
We can see it, Scott. Thank you.
Great. Thank you so much.
Do you want to put it in presenter mode? There you go. Great. Thanks.
Yep. Thank you. So thank you, Dr. Wallace, for that introduction and discussion about NF1. My name is Scott Plotkin, and I'm a professor of neurology at Massachusetts General Hospital. And I had the chance to look through the attendee list and saw names of many friends, including patients as well as other scientists and advocates. And so it's wonderful to have this opportunity to be on this webinar. So I'll take up the discussion now, focusing on NF2, neurofibromatosis 2, and I hope you'll forgive me for two things. Number one is some of the information will be repeated because there are many common themes between gene therapy for NF1 and NF2. And also, you'll have to forgive me that this is not going to be an in-depth discussion in the sense that we have some limited time here, but we're always happy to answer questions and to continue the discussion. It is important that I mention my disclosures that as part of my quest to bring therapies to patients with NF1 and NF2, I've had the great fortune of being involved in the founding of some companies focused on bringing therapies for NF1 and NF2. Those are potential conflicts that must be disclosed, and I do some consulting for AstraZeneca and a company called Missense. So the goals of this brief talk will be just to understand some of these basic approaches to gene therapy and really to frame some of the challenges and risks of this as it applies to NF2. So just as a quick reminder about NF2, it's about 1/10 as common as NF1, about 1 in 25,000 live births. This translates into about 10 to 12,000 Americans, and of course, many more worldwide. We know that it's caused by germline mutations in the NF2 gene. What that means is you're born with mutations in the NF2 gene. We'll talk more about that in a moment. I'll tell you now that if you want to be up on the latest language, we're not using the word mutations anymore. We're now using the term pathogenic variants. Pathogenic means disease causing, and so I'll keep the word mutation for now because many of us are familiar with it, but we will be eliminating that from our discussions. The diagnosis of NF2 is based on clinical criteria, and that is usually established by MRI scans, sometimes by surgery. And this involves the presence of bilateral, which means both sides vestibular schwannomas, or a single one-sided vestibular schwannoma, which are the tumors of the hearing nerve, and multiple other tumors associated with NF2. The average age of diagnosis is later than NF1, about 22 years of age. And we all know that these individuals affected by NF2 are at risk for multiple tumors and tumor types, some of which I have shown you on the right, including in the top left panel, vestibular schwannoma, those two bilateral vestibular schwannomas. On the top right, a characteristic meningioma on the internal portion of the brain. In the bottom left image, what we call a spinal ependymoma that has both a component that's white, meaning enhancing, and a component that's dark, which means fluid-filled cyst. And on the bottom right, a multiple level or multi-level schwannomas in the spine. And I'll just point out that these are predominantly limited into the central nervous system, but patients also get schwannomas outside the central nervous system, in the peripheral nervous system. Trying to switch my slides, so sorry. Let's see, is this not working? Okay. So really now to review much of the stuff that Dr. Wallace did, just to remind us, of course, what the genome is. You hear about the Human Genome Project, and it's just a reminder that genes are the genetic instructions in cells. And if you start with a human, if you look into the cell, within the cell there's a nucleus that contains the genetic information. And this genetic information is wrapped very tightly into 23 pair of chromosomes that include all of our DNA. And you can see here the DNA is made of this sequence, alternating sequences of 4 bases, CATG, that repeat in different varieties. And those that is what constitutes what we call the genetic code. And this is a nice visual representation of why we call it a genetic code. And we think that this encodes for about 20 to 25,000 genes, and that number keeps changing, usually on its way down in numbers. We used to think there were many more, but about 20,000 genes. One of those genes, of course, is the NF2 gene. We heard a little bit about that. Unlike NF1 gene, which is located on chromosome 17, the NF2 gene is located on chromosome 22. And you can see here that it's organized. Can you see, Simon, my pointer? Does that come across?
Yes, we can see it.
It does. So this is the anatomy of a gene where you have, it goes from the 5' end to the 3'. These areas that are in color are called exons because those are the areas that get made into the protein. And you can see here, if you look inside of an exon, you can see the sequence depicted right here. And as Dr. Wallace says, this is what gets encoded into a protein that actually has an activity. And in our case, for NF2, that protein is myelin. as opposed to NF1 where it was a neurofibromin. And just for comparison's sake, if I can get it, there we go. Just to show you a comparison in size between the NF1 and NF2 genes, the NF2 gene is about 25% the size of the NF1 gene if we use rough numbers. Whether you look at the RNA or you look at how many exons there are, which is the part that encodes for the protein, whether you look at the number of amino acids or the protein size, if you want to have a rough idea, it's about a quarter of the size. And that's important because we do believe that when it comes to replacing the NF2 gene, as you'll hear about, it can be packaged into for gene therapy, unlike the NF1 gene, which is probably too large. So these pathogenic variants, that is what we used to call mutations, can come in a variety of types. The most common encountered in NF2 are called loss of function variants. And what happens is you have a normal gene, as you can see here, which produces normal protein. When there is a pathogenic variant, or what they're calling here a loss of function variant in a gene, it can either be partially inactivated, so you can get a defective gene that doesn't work very well, or it can be fully inactivated where there's no protein produced. And those are together called loss of function variants, and that's most common in NF2. You can also see what we call missense mutations, and what this graphic is trying to show you is a bit of the genetic code as it goes from DNA on the top line through the intermediate stage of RNA and then to a protein. You can see in this case, this sequence of GGT encodes an amino acid we call glycine. So what happens when you have a substitution, so this G that's in red becomes an A, you end up not with a glycine, but with an aspartic acid. The details aren't important, but just to emphasize in this instance, what you have is a protein whose function is altered because an amino acid has been substituted, but it doesn't involve a complete loss of function of the protein. You have an altered function, but not a loss of function. These pathogenic variants, these missense mutations, are less common, but still present in a minority of individuals. And the last bit of information I want to introduce is the concept of tumor suppressor syndromes like both NF1 and NF2. And what is unique and special about these conditions is that individuals are born with one inherited pathogenic variant in every single cell of their body. And as Dr. Wallace says, it takes a second hit to take off that second pair of brakes that leads to a tumor. And what I want to draw your attention to is this idea that there's two types of tissues when we think of gene therapy. The tumors we all think about, those are cells that have loss of both copies of the NF2 gene in this case, but we also have a whole large number of cells that have one inherited pathogenic variant, and we also have to think what would be the effect on those, what we call normal tissues, obviously. So here are some of the different approaches to gene therapy for NF2, and I think this overlaps with what Dr. Wallace was talking about. Gene addition is what, in the top left panel, I will call gene replacement. So that's increasing the necessary gene. A second approach could be gene knockdown if there's a harmful gene. A third one could be gene addition. I use this in a different way, meaning you could put in some kind of suicide gene or therapeutic gene so that the cells that take up this gene cause that cell to die, or gene editing or gene correction where you're correcting the faulty gene. And I'll just emphasize that there are other ways. These are just 4 convenient buckets for our discussion today. I want to draw your attention to a fascinating example that we call proof of concept. The example of gene replacement for hemophilia, which is a blood condition where there is, the blood does not clot normally because of lack of a protein. And what this group did was give gene therapy to increase a factor we call factor 9. And what I want to draw your attention to, this is on the y-axis, the percent of normal factor 9 activity, and on the bottom x-axis is the time. And I'll just draw your attention of these 9 participants, all of them stopped spontaneously bleeding, meaning that their hemophilia was much, much better controlled. And why I wanted to show you this image is they only had between 10 and 40% of normal activity. And so in some conditions, we don't know yet about NF1 and NF2, partial restoration of gene function may have an effect. We don't know that, but in other conditions that has been the case, and that is partly of what we're thinking about when it comes to gene replacement therapy. So as Dr. Wallace also mentioned, and we're going to hear about more from Dr. Marks, safety concerns as we enter the gene therapy is incredibly important. So here are some of the concerns. What happens if we get overexpression or overproduction of the target gene, particularly in a condition with a tumor suppressor gene that is stopping cell proliferation or growth? What about the potential risk of viral vectors? These are the Trojan horses that deliver the gene therapy into the individual. And what about short and long-term risks? Certainly, we can't just monitor people for a year or two. These conditions where you're changing the gene structure of a person really require very long-term monitoring. So when we boil now down to NF2 gene therapy and we think about these four options, understanding there are more, when we think about gene replacement, the questions that come to mind immediately are how much replacement is necessary? 10%, 5%, 50%, we don't know. The corollary is, what if it's 150%? What deleterious or negative results would that cause? What does it mean to do gene replacement on normal tissues versus tumor tissues? When we think about gene addition, we don't know yet for NF2, what are the best genes that could act like suicide genes that could kill NF2 tumors? When we think about gene knockdown, this is really important for cancers that have overproduction of genes that cause growth. Not clear how we're going to use that in NF2, where we're worried about a lack of a gene product. For gene editing, to correct the NF2 gene, of course, this seems like the holy grail, something we're all interested in, but that technology is maturing and is not quite there yet, because we worry about what we call off-target effects. It corrects the NF2 gene, but also may make changes in other genes that are related to the NF2 gene. And finally, for all of these approaches, as I began to mention, what about the effects on tumor versus non-tumor tissue? And very practically, how do we deliver this to the cells of interest? What's the best method to deliver it? Is it by mouth, by vein, directly into a tumor, directly into another tissue? And I think we're going to be touching on some of these issues with Dr. Marks soon. Outside of these technical issues, I cannot stress enough that we as a community need to begin tackling the ethical issues surrounding this. So once our laboratory colleagues say, this is what we should do, this is the gene therapy we should look for, I believe that our community needs to answer some important questions. Who are we going to treat? How old will they be? Will we go for young patients with severe disease or older patients with mild disease? Who's the right group? What are the right tumor manifestations to target? What's the stage? And most importantly, what do we call success? For hemophilia, it was pretty straightforward. That was a lack of spontaneous or unprovoked bleeding. And NF2, it's a little less clear, and we need to discuss this in more detail. So with that, I think that we're very fortunate to have Dr. Marks, who's going to bring in some of the context in which we're developing these therapies. And I think we'll defer the questions to the end. Thank you very much.
Great, thank you very much. And let me just get here to be able to share my screen. Okay, hopefully that is, we'll share in a second.
Yep, Dr. Marks, we can see your screen if you just want to go into presenter mode.
Yep, it's being a little temperamental at the moment. Let's see if I can make that happen. There we go. Okay, thank you. So thanks very much. So what I thought I would do is just tell you from the FDA perspective a little bit about how we think about gene therapy, its development. And I'm going to apologize that I will duplicate a little bit here of what's been said, but it hopefully will put things in context. So from our perspective, gene therapy defined as the modification or manipulation of gene expression or alteration of the biological properties of living cells for therapeutic use. And in general, that means that any product that mediates its effect by modifying how cells perform some of their functions, either how they replicate when they replicate their DNA, or how they make proteins from their DNA through RNA, by altering those things, they fall into gene therapy. And so a variety of gene therapy vectors, things that make gene therapy happen, include things like nucleic acids, such as plasmids that might be put into cells, genetically modified microorganisms, viruses, which are often used as a delivery mechanism for gene therapy, but also even bacteria could be forms. There are now certain types of genome editing technologies where one can almost like a typewriter correct single base pairs within our genome, and we regulate those. And we also regulate, as gene therapies, cells of the body that are removed, genetically altered in a culture dish, and then put back into the body. In terms of our authority in this area, we regulate gene therapy in both the area of changing things in the cells of the body that one doesn't inherit from one generation to the next, as well as heritable genetic modifications. I will say that at this time in the United States, all of the gene therapy that's allowed to proceed is just changing the genes in an individual during their lifetime. At this time, Congress does not allow gene therapy that would be heritable, that would be able to be passed for one generation to the next. You already heard about the fact that there is a gene therapy for hemophilia getting close to being a reality with an approval. There are currently 4 approved gene therapies in the United States and 5 approved gene therapies in the European Union. Some of these are obviously overlapping, and there are 2 of these genetically modified cellular therapies. That's where the cells have been removed from the body, the gene therapy has been performed, and then they're given back. Those are 2 products that are used for cancers. And there's one that's used for into the eye for a type of blindness that's caused by a genetic mutation, a genetic disorder. And that's Pheretogene, and one that's caused by another pathogenic variant, to use the more appropriate term, without falling back into the older term, Ansemnogene, which is for spinal muscular atrophy. And that's a product that's administered systemically that has had some pretty spectacular results. The European Union has an additional gene therapy in addition to those for beta thalassemia, which is a blood disorder in which people don't make enough hemoglobin and therefore don't make enough red blood cells. So at FDA, we're responsible for making sure that products, including gene therapy products, are both safe and effective. And that means that they have to meet a legal standard for efficacy. Unlike certain parts of the world where regulators only get involved at the very end of the process of product development when the licensing applications are submitted, at FDA, we're involved in the process of product development throughout the life cycle, from concept of products through post-marketing and surveillance. And in fact, an increasingly important part of our mission at FDA is to facilitate the development and approval of innovative products that address unmet medical needs. So typically, just to give you an idea of the difference between typical research and the research that's done, clinical research that's done for products that are for gene therapy, normally for typical drug products that might be for blood pressure or to help one sleep or to deal with other disorders, you go through an orderly set of testing in which the initial testing is done in a relatively small number of people just to find out if the drug is safe and to figure out the right dose to use. That's phase 1. It then moves into phase 2, which looks at the initial efficacy and side effects from the drug. Phase 3 is when then one gets definitive efficacy and looks at additional information on side effects. And then phase 4 comes after a drug is approved. but you'll notice the numbers in parentheses. Usually phase 1 is up to about 100 people. Phase 2 is maybe up to 500, and phase 3 trials can be 10,000 patients, and for vaccines, it can be actually even thousands more. But when you think about it, for gene therapy, the trials are not anywhere near this size because they can't practically be, because many of the disorders that are being treated have much smaller populations. And so for relatively uncommon diseases, clinical trial programs are developed to be fit for purpose and depend on the factors such as the strength of the effect that's seen and the ability to measure outcomes. So if there is a very strong effect, that is, if one can show remarkable things like someone is not able to make blood, you give a gene therapy and they're able to make blood, one doesn't need to treat a remarkably large number of people in order to demonstrate that one has efficacy. On the other hand, if one has to see a change in some function over time, it can be a little bit more challenging, and then the number of people in a study might have to be larger. But either way, in general, for rare diseases, we see things where a phase 1-2 study of initial safety and dose finding is usually just done in a handful of patients, somewhere on the order of 5 to 20. And then the studies that actually lead to product approval are generally only done in several dozen patients, or perhaps up to 100 in total in the program. So it's a little bit different for rare diseases. What we're trying to do at FDA is obviously advance gene therapy as much as we can. And we do that by using certain programs called expedited programs, which allow us to help developers move things through the process more quickly. We give out regulatory guidance. We meet with sponsors of products to help them figure out how to develop things. We help with advancing manufacturing technology, and we develop new regulatory policy. And I may give you an example of our new regulatory policy that we're actually working on right now, so you can see that we're trying to address issues that are coming up. So our expedited development programs, just briefly, just to give you an idea, many of these have been put in place to help sponsors be able to get answers to requests to FDA in a more quick amount of time than they would be otherwise, or they allow one to get an approval based on some intermediate endpoint rather than a definitive clinical endpoint. And more recently, breakthrough therapy and regenerative medicine advanced therapy designation have been put forth for very promising products to allow the agency to help sponsors move things as quickly as they can through the development process. The RMAT designation process applies to gene therapies, and it was put in place so that gene therapy products could get additional, that are qualifying for this program because they show promise, can get additional attention of the agency. What we say when we're saying about guidance documents, the guidance documents, when you hear us talk about that at FDA, what we're talking about is really advice for people developing products on how they can best do that and stay in compliance with our regulations and ultimately produce a high quality product. We've put out a variety of guidance about how to manufacture these products, how to test them to make sure that they're, in certain cases, are not features of the products that could be harmful. We have some guidance on how long you need to follow someone after you give them a gene therapy, because as was already alluded to, one of the real concerns with gene therapy is that we have to make sure that they're safe. And this is particularly challenging in certain diseases. And I'll mention one in a moment, but you can see that we have 3 clinical guidance as well in this list, 4, 5, and 6 here: hemophilia, retinal disorders, and rare diseases. Hemophilia has already been mentioned, and that's a good example of one of the challenges in the gene therapy area, because just because something is a gene therapy doesn't necessarily mean it is that it is better than an existing therapy. It means it's just different. In some cases, for instance, for hemophilia, there are existing therapies that some of which are quite good. And so we have to really carefully think as we're developing products, whether gene therapy makes sense or whether gene therapy at this point in time makes sense. In some cases, what I mean is that it may be that ultimately when we've done a lot of work with gene therapy and have a highly effective product, it might be clearly head and shoulders over an existing therapy. But in some cases, it may take time to get there. I'm not saying that necessary for hemophilia. I'm just saying that overall so that we have an idea that just to remember that just because it's gene therapy does not necessarily mean that it's definitely better than existing therapies. It just means that it has differences and has the potential to offer benefits. So finally, I just want to just take like 3 minutes here and tell you about a piece of policy that we're working on. I think is of interest to people with about how we're trying to deal with this issue of relatively uncommon disorders, because ultimately, when we start to think about rare disorders, many of them break up into even more rare disorders. And what do I mean by that? Well, there are uncommon disorders that ultimately, when you look at the pathogenic variants that comprise them, the changes in the DNA, it's not just one change in DNA that gets you to that disorder. There are a whole host of different ones. And so if one starts to think about fixing those different mistakes in the DNA, one actually has to have slightly different products. And that brings up this kind of new regulatory concept of individualized medicine. You may have heard of personalized medicine, which is finding the right drug off the shelf to treat the patient. That's when you look at some characteristic, a laboratory test in a patient, and you figure out, oh, this drug seems to fit that patient. On the other hand, there's individualized medicine, which is about creating the right drug to treat the patient based on the characteristics that you observe in the patient. Now, individualized medicine ends up being able to be further divided into the kinds of products that one needs. There are products which we would consider customized products, in which one has products that all have the same indication and the same mode of action, but might be slightly different. The example I put on this slide is about personalized cancer vaccines, but you could also just as easily imagine that if you had different genetic mutations, or sorry, I keep saying that, sorry, I should really stay in the personalized variant. But if you have different variations that cause a disease entity, that is, and you have to correct the different ones, those are all essentially acting for the same indication and really by the same mode of action, and they might fit under this customized products. On the other hand, there are created products where you have to make a different product for instance, different diseases, because you might have just wholly different lysosomal storage disorders or other genetic diseases. And so the way we think about this, just to be a little bit of light relief, since we're all kind of ready anyway. So the difference here of personalized medicine is like ready to wear. These kind of customized products are made to measure, and these created products are like bespoke tailoring. The reason why it's so important is because there are so many thousands of rare diseases, and we need to find a way to efficiently be able to address those. Now, gene therapy is particularly helpful here in rare disorders because one can't take the time to make a small molecule per se. One might not be able to make a protein therapy in big bioreactors, but if one could quickly address the issue by developing a gene therapy construct, that could be very helpful. So we're committed to trying to help move this forward. And in order to do so, we're going to have to find ways to leverage data so that one doesn't have to go through a big preclinical development program, that is, develop all the information necessary before you can treat a human every time for each new construct. And so we'll need to leverage data as we develop things, and we're trying to figure out now how to do so, and we have some demonstration projects going on. We also will have to figure out how to understand with very small populations of patients how one figures out whether something's really working or not. And to do that, we'll probably use some modern statistical methods like Bayesian and other designs to try to understand that. So just to finish off, we're very committed to advancing the development of novel therapies for populations of all sizes, and that includes helping throughout product development, and whether it be from the concept to manufacturing to the clinical trials. But ultimately, what's very important to us is making sure that when the final product is there, it has to be safe and effective. It has to be something that patients can feel comfortable taking, and that it really brings clinical benefit. And I'll stop there.
Great.
Thank you. Thank you so much, everybody. What I've been doing, first of all, amazing talk. I got a lot of texts and messages, private messages, that people were very enlightened by this panel. So thank you. We've received quite some questions, and being mindful of time, I wrote a couple of them that kept coming back. So there is one question: if the gene therapy is not perfect, could that be harmful?
So this is Peter. I can start by saying that I think the concern here is, obviously there's a lot of different ways that could be answered, but I'll say the most simple one that I can think of, and then maybe the other panelists may comment, that rather than being necessarily harmful in terms of causing an adverse effect, which is obviously something that's possible if it's not effective, because anytime you give gene therapy, there is the potential that it could cause some damage to the rest of the DNA of the body. But aside from that, one of the concerns we have is if one gives a gene therapy that's not effective using some of our current gene therapy vectors, one can mean that someone may not be able to get a gene therapy using a different construct and that same gene therapy vector. So ideally, one wants to try to have vectors that actually are effective and do the job. But it is true that gene therapies can be associated with adverse effects, and that's why we want to try, before we see them used in humans, to make sure that there's good evidence that they're going to have the benefit that they promise to have. But maybe the other panelists want to add to that.
Well, I think this is Scott, and I think that's exactly right. And the only thing I would add to that is I was alluding to this discussion that our community needs to engage to. The community being patients, families, physicians, laboratory researchers, foundations, to understand, like any therapy with risk, how do we start that's reasonable? And so, yes, there are risks. The word we like to use is we like to mitigate risk, which means to minimize it. And we do that in many, many ways. Number one is we want the best preclinical data before moving something into patients, and then a group decision on who are the right patients to move into. And I'll just put a plug. The RAINS initiative, which is the Response Evaluation in Neurofibromatosis and Schwannomatosis. It's a volunteer organization of researchers and patient representatives. Our December meeting will focus on beginning this discussion. It's a long discussion, but we're going to initiate it the first Monday in December, and we're going to try to include many people. If we're lucky, we'll get Dr. Marks or one of his colleagues to join us. We'll shoot the moon there. But either way, we know it's an important discussion to have.
So there was one question talking about preclinical models. There was a question about the use of which models, right? The use of mini pigs, the use of mice, the use of all these models. So I don't know if the panel can comment on the models or the experience with what we need before we feel confident that we can go to a patient.
Dr. Wallace, would you like to do that, or would you like someone else to?
Yeah, for NF1, I think we have so many different pathogenic variants it's actually hard to necessarily have a model for each one. But I think in some cases the therapy is going to dictate what the best model is going to be in order to test it and make sure that it's going to work. And it's, in some cases, it's always going to start out with cells. And in many cases, it's going to have to move to animals of some sort. And I just think there's not going to be necessarily one model that's going to work for everything for NF1.
I think the one thing I would add is, I think for much of the community, the safety of these models is determined by what we call non-human primate studies, which are animal models of non-human primates, which is always a challenging position for many of us to think about. But in fact, in order to really make sure that these therapies are safe for humans, that has been the standard for many of these conditions, I would say. I don't know if Dr. Marks would like to add to that, but only if he wants.
No, I'll leave it at that, so we have time for any more questions if need be.
Of course, there is the $10 million question. How far are we away from NF1 and NF2 treatments for patients? There were multiple questions that were talking about that, so I don't know if anyone wants to comment on that. And kind of also question if there is already some commercial entities working on NF gene therapy. So kind of a little bit of how does the landscape look? Is there hope?
I think what you're seeing now is we finally have the tools and the regulatory process to make forward movement on these areas in ways we couldn't before. And at the same time, I would argue from my perspective, which is a personal perspective, we're still at least a decade away from having effective gene therapies for NF1 and NF2. Now, that doesn't mean that there's not going to be a lot learned along the way, but I do think that there are many unique things about these conditions which are different than hemophilia or retinal disease or the other things that we talked about. And that's why I think all of us have been interested in developing of what I call a portfolio for NF1 and NF2. That is, gene therapy is something to work actively on at the same time working on drugs like selumetinib for NF1, bergatinib for NF2, and that this forms an important part of our portfolio as a community that has to be pursued, but realistically, in my personal opinion, it is not a near-term likelihood.
Yeah, this is Peggy. I think the only gene-based therapies that might potentially come to fruition before that would be the ones that would not be a permanent fix. It was something that you would have to take routinely that would alter how the gene is working or how the RNA works in order to get the bottom line effect of having some normal neurofibromatosis. So those are basically kind of a drug type of a system. And so it's possible those could come to fruition sooner, but the true gene therapies are definitely years away, many years away.
Yeah, you also, Peggy, you also mentioned that there is an enormous number of mutations in NF1, right? A large gene, high number of mutations spread across the whole gene. Is that
Right.
a complexity that I think we should recognize, right?
It's one of the complications related to gene therapy, and also just understanding the mechanisms of how things work. It's possible that there actually could be some NF1 pathogenic variants that aren't just simple loss of function. And if that's really the case, then that requires a different strategy to try to fix that. So there's a lot of different mutations, the old system, scattered across the gene, but the gene has discrete 58 exons. So you could potentially, as one of our groups is developing therapies based on actually fixing everything in that exon. And so there's targeting going on at all different levels. And it may well be that depending on exactly what is happening in a given patient, there may be one tool versus another that's going to be better.
I want to be respectful of people's time, but there was one burning question which I think would be, oh, yeah, would be interesting to throw out there is what happens. Let's say we correct the NF1 gene. Do you expect, for example, cutaneous neurofibromas and plexiform neurofibromas to shrink? Or do you avoid them to grow? Or what is kind of the end point, let's put it this way? What are you looking for?
Well, we would hope that the tumors would shrink. I mean, based on the new FDA-approved drug, when you fix that imbalance, the tumors often shrink. So maybe if we get NF1 restored, the tumors might stop giving themselves growth signals and actually not only stop, but potentially shrink. Certainly it would potentially should prevent any new tumors from forming, and certainly prevent those that are already there from getting bigger. But we don't know for sure.
The only thing I would add is that's why the CTF, the Department of Defense, and many other organizations have spent millions and millions of dollars creating these animal model systems that we hope will give us a very good idea. I think that's where we are now, is trying to see, does it reverse tumor growth, meaning shrink it? Does it stabilize it? And the investment that we've put into creating these model systems is going to pay off as we move forward in gene therapy.
I don't see a lot of other. Are there, that's an interesting question as well. Are there neurofibromin analogs that have the same function?
There are no other genes that are copies of NF1. There are some genes that are partial copies of NF1 scattered around the genome, but they don't work correctly in terms of making neurofibromin. So the basic idea is you've either got to fix neurofibromin, replace it, or you've got to fix the downstream pathways that are being imbalanced when it's not working right.
Which is what we are doing with drugs now mainly.
Mm-hmm.
So there are two people that have raised their hand. So Carly Kegel and Barbara Franklin. Simon, would you mind unmuting these two people just to see?
Sorry, Carly was first. I'm going to allow Carly to speak, and then Barbara will get to you next. Go ahead, Carly.
Hi, my daughter has NF2, and so does her dad. Her dad actually saw you a year ago, Jeff Rosado. I don't know if you remember him or not, but my question is, you know, the age, like her NF2 seems to be progressing a lot faster than his, and so I know you guys don't know the types of patients yet that you would like to, you know, use for the clinical trial if you had one. I guess my question would be, you know, would there be a specific type of, like, what would be the things that, like, you would look for in a patient? Is it certain types of tumors they would have? Would they have to have more, you know, meningiomas versus schwannomas, or do you know that kind of stuff yet?
Let me reflect the question back to you. What do you think we should do? I think that's the right question. Do you think it would be better to treat an older person who was much sicker, let's say couldn't walk well because of balance problems? Or would you rather treat a young person who was less affected, maybe neurologically doing well, but has many tumors that were growing?
I think that it would be better to do the younger patient, just because, well, for a lot of reasons. I mean, you know, Jeff, her dad, has obviously a way more progressed, but not as fast as hers, but she has a lot of meningiomas in her brain that are going to potentially have to be removed in the next few years. And, you know, if I could save that, then you're saving a life rather than somebody who probably is already going to, you know, not.
So what if I told you that you only get gene therapy once in your life because of the reasons that Dr. Mark said? It has to do with the immunogenicity, or the immune response to these particles that bring it in. Would you still treat with the early virus vectors, or would you not? So these are the kinds of we don't have to answer on the phone. What I want to bring out is, I don't think it should be the doctors. This is my own personal opinion. It shouldn't be the doctors who decide this. I think we need to engage in a conversation with patients and patient representatives and their families, because if I wish I knew all the answers. I think what I've learned in 20 years is I don't know. And Edith, Garrett, many other people along the way have really taught me a lot. So yeah, I think it's a good question. I wish I could tell you the answer.
And we're going to move on. Go ahead, Peggy. Sorry.
Oh, sorry. I want to add something to that. And this is a plug for, keeping our federal research funding system healthy. One of the things that are being worked on in terms of gene therapy are developing some of these vectors that are not going to be immunogenic, or that there's going to be systems developed that they'll be able to accommodate for that, so that you could get retreated. So there are scientists working at every level of gene therapy all along the way to try to solve a lot of these problems. And it's really exciting work, and we've got great tools. We just need to keep up the funding of the labs in order to keep moving forward.
Yeah, I completely agree. And I think it's like what Scott said, right? It's on the border of treatment and ethics, right? What is ethical to do? And it's not as clear. It's not, there is so much advancement happening still, that I think it is indeed a tough question. Barbara.
Well, thank you to the entire panel. Whoa, my picture showed up. Thank you to the entire panel. You guys are incredible, and during these very difficult times to find the time for us is very appreciated. And to Carly, I say, my son is in his 30s, and I've been where you are. And I will just share that I remember Mia McCullen, when Adam was first diagnosed in NF2 research, said, the big concern is don't chase the next shiny object. Don't make him a guinea pig, because it's quality of life, and you're seeing gene therapy progressing. So, if you get one shot, be very careful about when you exercise that one shot, because I've certainly, with Adam, been guilty of trying a couple things that I wish I hadn't. So that said, my question is, with all the different mutations, and I'll go to NF2 because it's a smaller gene that we're dealing with, but with all the different mutations, will there be all these different gene therapies? And therefore, in my son's case of a splice site, will that be a very specific one that he couldn't use a missense mutation one, for instance?
So, this goes to the question that Dr. Marks was bringing up of individualized medicine and personalized medicine. And I think the next 3 to 5 years is going to shed a lot of light on this. I think with investment from foundations and companies and the federal government, we're going to really learn a lot. To be clear, Barbara, your question is really spot on. And what I mean by that is our animal models that we've invested in over the last 2 decades do not recapitulate human disease with its same variety that we want for gene therapy. And so your question is so spot on. It's really going to be, how do we tackle this genetic variation across individuals in our clinics? And we're aware of the challenge. We're working on the challenge, but I don't personally have an answer. I don't know if Dr. Wallace has a better answer than I do, but it is, I think, the right question. One of the right questions.
Thank you.
Okay, with that, first of all, Dr. Marks just apologized that he had to leave because he had another meeting. We're 12:11. I really want to thank the panelists indeed for the time they dedicated in this COVID craziness. So thank you, Scott. Thank you, Peggy. As always, you're always ready to help. And
now I can put back on my mask.
Scott's going back to clinic. He's in clinic today. So thank you so much. Also, of course, thank you to Dr. Marks. This was an amazing panel. And I hope it has helped people to really understand that although gene therapy might be announced in People magazine and newspapers that it's easy, it is. We just wanted to make sure that you understand that it's not a cookie cutter yet. So with that, thank you so much, everybody, for joining us, and looking forward to see you soon.
I'm going to let the marketing guy jump in just for 1 second before you'll hop off. I'm sorry. It is May is Awareness Month. We just want to make sure you're aware of some other activities taking place later this month. May 17, World NF Day is this Sunday. Wear blue and green. It's for all forms of NF. And then May 22 is NF2 Day. Wear green and blue for NF2 on May 22. We'll also be hosting a special webinar that day, which you will, for on both NF2 and schwannomatosis, which we'll be announcing very shortly. You will not want to miss that. Another 30 seconds, shine a light on NF. You all know about this. Buildings will still be lighting up around the world, even in this COVID craziness. We know that Niagara Falls will be lighting up, so be looking for announcements for that. We have pivoted. Home is where the heart is. We all are at home, and people are finding ways to light up blue and green in their homes. So go to our website for that. And please don't miss our Make NF Visible photo series, which shows and has paired together people with NF of all forms, both visible and not so visible in terms of the things that you can see. And then very important, this Sunday, May 17, Home is Where the Heart Is, a Zoomathon, which is a new, updated, fancy, modern era telethon to benefit the Children's Tumor Foundation. Will be on Facebook, YouTube, and on Zoom. And if you go to our site, ctf.org/heart, you will see an amazing number of celebrities. I'm not going to read them all here, but you'll see lots of famous names: Jesse McCartney, Emily Deschanel, Emily Osment, Martin Short, Dave Thomas. Just going to skip ahead: Alec Baldwin, Paul Schaefer, Bob Saget, Danica McKellar. Visit the site. You'll see it first time that this many really famous people have come together. Athletes, Top Chef, Top Magician, America's Got Talent winners. Missing on this page, because he just joined us, is John Stamos. So we actually have the full house cast: Bob Saget, Dave Coulier, and John Stamos. So again, this Sunday, ctf.org/heart to learn more, or you can right now pull out your phone, text CTF Heart to 51555, and you'll be added to the list to receive notifications. So sorry, I had to interrupt our important webinar today, but just to make sure that you all know that this is happening this Sunday. And with that, I think that's it. Back to you, Annette.
Okay, thank you so much. It's just the strength of a community, and as Peggy said, we can end NF, but we need to be together. We need to stick together to get the job done. So with that, thank you so much for being with us, and hope to see you very soon, maybe on Sunday. Have a nice day.